Good afternoon, and welcome to Radcliffe Institute. It gives me great pleasure to present Dr. Luhan Yang today, who will be talking about extensive pig genome engineering to transform xenotransplantation. She is the co-founder and CEO of QihanBio. She's also the co-founder of eGenesis. She is leading the international team to deliver human transplantation, tissue, and organ using gene engineering technologies. She previously developed the highly programmable genome engineering tool, CRISPR-Cas9, for use in mammalian cells, and pioneered the first isogenic human stem cell lines to model human diseases at the tissue level. She was recently honored in Bloomberg 50, named the Young Global Leader by the World Economic Forum, and was among the 30 under 30 in science and healthcare by Forbes, and was a laureate at the Bloomberg 50 in 2017. Dr. Yang holds a BS degree in biology and psychology from Peking University and a PhD in human biology and translational medicine from the Harvard Medical School. Thank you, and please welcome Dr. Yang.
Good evening, everyone. Thank you so much for coming on Friday night. And thank you, the organizer, for inviting me to be here. It's truly an honor. So today, I'd like to share with you how we are using gene editing tool to deliver human transplantable tissue and organ by using pig as a bioreactor. So first, I'd like to introduce you to QihanBio. So we are a sister company to eGenesis, which I also co-founded in China. And we have a shared mission to create a new world where no patient should be died by waiting for organ transplantation. I'd like to first acknowledge my team. So we have a seamless team in both China and in Cambridge, about 80 people. We are not only aspired to create organ for human transplantation, but also to create a novel business model that we can leverage the best of both worlds to address a global medical issue. So we always know that organ shortage is a medical problem, but the delta between the organ donor and the people who need organ transplantation is daunting. So in this country per se, despite the fact that there are over 190,000 people are waiting for organ, there are only about 70,000 transplantations each year. So patient is dying. If we do a mental exercise, how we can address this delta? So the first one is we can encourage more organ donation, but the number has been stagnant for the last 2 decades. And the second one is maybe we can use stem cell and 3D printing to create an organ in the dish. And the approach we are taking is we try to harness the organ the Mother Nature has created for us in an animal, but change in the way that human compatible. So our vision is by doing so, we can create unlimited supply of the organ for the patient. Our near-term goal is to make the pig organ as good as human organ, but in the future, we have the opportunity to change the organ as good as identical twins organ so that we don't have to allow the patient to take immunosuppressant all through their life. And we regard it as a platform that the pig can deliver a big spectrum of product ranging from cell therapy to organ transplantation. And the concept of xenotransplantation using pig organ for human transplant is not a novel idea. Since 1960s, before human-to-human transplantation, the pioneers in the transplantation domain, such as Dr. Tom Stuzzle, started to try the pig organ to human transplant. And this is a clinical trial done at the Tulane University in the 1960s. So in this clinical trial, 13 patients with kidney failure were transplanted with a monkey kidney. And here we are looking at the medical profile of the patient survived the longest, over 90 days. And we can see that with the organ transplantation, the patient start to produce normal urine, and the creatinine and other biomarker are normal until over 3 months, and the patient unfortunately died out of the electrolytes imbalance, likely due to the organ rejection. In the 1990s, with the invention of the first generation immunosuppressant, people started to look at xenotransplantation again, and this was a trial done by Genzyme at the 1990s by putting the pig neuronal cell into the Parkinson's disease patient. And we can see that after one biopsy of the patient brain, the clear existence of the human neuronal cell as well as the integration into the system. However, there are 2 problems. First one is, despite the fact that in the trial, the patient were transplanted with 10 million pig neuronal cells, after a couple of months, only about a couple of hundred cells survived, meaning that even for the brain, which is quite immune privileged part, there's still a strong rejection to the pig tissue. Because of that, there is no clear evidence that it's effective for those Parkinson's disease patients. And also in the 1990s, people discovered that HIV was passed from monkey to human, and by that time, a new group of virus called porcine endogenous retrovirus was discovered in the pig genome that potentially can be passed from the pig cell to the human cell. So it didn't work and potentially can create new epidemic diseases. Due to the concern, FDA and WHO shut down all the clinical trial, and there had been a silence in this field for the last 15 years until now. So at QihanBio at eGenesis, our vision is by using gene editing to address the 2 fundamental problems of xenotransplantation. So on one hand, we want to modify the genome in a large-scale fashion to restore the immune and functional compatibility. And on another hand, we want to eradicate those porcine endogenous retroviruses from the pig genome to address the viral transmission concern. And the time started when I was a graduate student with Dr. George Church at Harvard Medical School in 2013. And I worked with my collaborator, Dr. Prashant Mali, who was a postdoc and now a professor at UCSD. So inspired by the paper published by Jennifer Duna's lab, we wonder whether we can humanize the CRISPR system in the human cell and to see whether it can cut the genome and enable gene editing. And we first demonstrate that the tool can incur robust cutting. And another experiment we did by that time is we wonder, because CRISPR is a 2-component system, the enzyme and the guide RNA, which dictate where the enzyme cut, whether we can feed the system with multiple RNA so that it can cut multiple sites at the same time. And we chose 2 sites to target. You can see the red and green. When we only deliver the red RNA and the guide and Cas9, we can see insertion deletion in this particular region. In similar case, when we deliver the green RNA and the Cas9, we see small insertion deletion in this region. However, once we deliver both RNA with Cas9 together, we can see a larger insertion deletion in this region, which gave us the vivid evidence that the system is multiplexable, can target multiple places at the same time, which gave us the courage that maybe by using the beauty of the multiplexability, we can start to tackle the problem of xenotransplantation. As a scientist, we believe what we see. So the first experiment we did is to see whether there are truly viral transmission across the species. To test that, we mix the human cell together with a pig cell for 1 week. Then we isolate the human cell and to see whether there are any viral particle. And in this picture, you can see that with a human 293 cell co-culture with a pig cell for 5 days and 7 days, we start to see the pig viral element. And there are different serotypes of virus, and we determined that all of the viral type will have the transmission activity. We keep the infected human cell for 4 months, and we found the viral titer increase. So we wonder that whether this pig virus gained the new infectivity in the new host. So we co-culture the infected human cell with normal human cell and to see whether they can jump horizontally. And we found that the fresh human cell with encounter of infected human cell also can carry the virus after 1 week of co-culturing. Indicate this, the in vitro, at least in the in vitro setting, the PERV, the pig to human infectivity and human to infectivity is real, that we have to address that. Knowing the problem, we sequence the whole genome of the pig, try to find the location, copy number, as well as the sequence of those viruses. And we found 62 copies of them in the pig genome. So here we are looking at the genomic structure of the porcine endogenous retrovirus PERV. So it's about 7,000 to 8,000 base pair sequence with 3 genes. And the essential gene is the PAW gene, which encodes the reverse transcriptase, which is essential for viral replication and infection. So we designed CRISPR-Cas to target the conserved region among those 62 copies of virus and inactivate all of them to show that we can eliminate the activity. With a modified cell, we also wonder, because in our human genome, there are also endogenous retrovirus, and whether our viral activity can resurrogate the pig virus. So to test that, we overexpress the human version of PAW gene into the modified cell, and we didn't see any signal after a couple months to further validate that it's pretty safe after we get rid of the endogenous polymerase reactivity of the PERV. And in collaboration of Dr. Luhan Yang at Yunnan Agricultural University, and we take the modified cell, clone a pig out of it, and share with the community 2 years ago. This is the first pig of its kind without any endogenous retrovirus. And I'm happy to share with you guys today that now the pigs are 2 years old. They are pretty happy, healthy, and they are fertile. We also determined the genomic modification can be passed to the offspring, which really set up the good foundation for us and indicate that at least for the endogenous viral transmission part, it could be addressed through our platform. To eliminate virus is one thing. Another puzzle we have to solve is how we can create the immune compatibility. This is how we approach the problem. So we imagine if we put a pig organ into human body, what would happen? So in the first phase, we imagine that the preformed antibody in our body will recognize pig organ as foreign, then trigger rejection through the antibody binding and complement toxicity. Later, the coagulation incompatibility between the pig organ endothelial cell and coagulation factors will trigger a cassette, then lead to the vascular injury. And there are other immune cell mediated rejection we have to consider, including NK cell, macrophage, and T cell. And there are, of course, adaptive immunity and inflammatory reaction. So our strategy is to understand underlying each bucket, whether there are some molecular pathway has been identified to eliminate the xenogenicity and also some pathway to dampen the human immunity. And this is what we have now. So the table we are looking at together is the transgenic pigs people produced over the last 2 decades, try to address different parts of immunology, including antibody binding, complement, coagulation, and cell-mediated rejection. And each number is a de-identified gene identity. So that I want to bring your attention to this pig from Munich. So they have 3 modifications. The first one is to get rid of the largest antigen, which is a glycan, only expressed on the pig cell surface, not human. So it's the most dominant antigen. So the Munich pig first get rid of that largest antigen, and the overexpressed 2 human proteins, CD46, which is an inhibitory factor to inhibit the complement cascade, and also the thrombomodulin to inhibit the coagulation dysfunction. Over the last 2 years, we created our Model 2 and Model 3. Model 2 is the pig with modification for immunology, and model 3 is a combination of PERV knockout and immunology modifications. And our model 2 and model 3 have more than 10 modifications, including the 3 modifications the germline group has. And once we have the pig, the first thing we want to look at is whether those genetic modifications really translate to proper protein expression in vivo. So we look at those transgenes we want to get rid of are truly negative in our modified pigs. And for the human transgene we want to express, they are negative in the wild-type pigs, but are highly expressed in our pig 2.0 and 3.0, really indicate that genetic modification not physically present in the genome, but also translate into proper protein function. Having the modified pig, then we want to test whether those genetic modifications are doing what they are supposed to do to confirm some immune invasion into the human immune system. The first experiment we did was to incubate the modified cell with 200 people's serum, then to see whether there are some antibody binding. So the red bar is the wild-type pig, and 2.0 and 3.0 is our modified pig, and the HUVAC is a human cell as a control. So we found that with our modification, we can significantly reduce the preformed antibody binding in a human population, and, but there are still about 5% residue binding above the human level. We don't know what does that 5% mean in vivo, which we are doing some in vivo experiment to have a better understanding. And we are also trying to search for additional antigen, try to fill the gap. The second experiment was the toxicity assay. So we put our modified pig cell in the human serum, then add the complement factor. So if it's a foreign agent, then the whole system will kill the cell. And we found the same case for the wild-type pig. So there are tremendous cell deaths, but our pig 2.0 and 3.0 has same level of resistance to this human immune attack, similar to the allo level, which give us a confidence that maybe we don't need hundreds of modifications. Our modification can at large address the hyperacute rejection. And we also did similar assay to look at human NK cell attack to our modified cell and found similar immunity, and so as a human macrophage attack to the pig cell, and found the modified cell has same level of protection as a human cell. And the last experiment is we perfuse the pig organ with the human blood to see whether there are some functional incompatibility which could address the thrombin formation, abnormal thrombin formation, and we found our modified pig restore the coagulation function. Of course, the golden standard is to put the pig organ into a non-human primate as a surrogate system to understand more about the in vivo immunity. Experiment is going on in both this country and in China for us to have a better understanding of safety and efficacy. But we truly hope one day a pig can save 8 patients. So we can deliver liver to the liver failure patient, the pancreatic islet to the diabetic patient, the kidney to the dialysis patient, and the heart for the heart failure patient. And we envision that this is a platform. So our first stage is to address the organ shortage, and we hope to improve the organ better than human organ so that the patient do not have to take a lot of immunosuppressant. We also think it's a platform for us to further enhance the function of the organ. For example, we can engineer the liver to be cancer-free or virus-free so that without touching the human germline, we can transplant an organ not only compatible with human body, but with enhanced and novel functions. With that, I'd like to conclude. So I hope I convince you that among the two, between the two major issues of xenotransplantation, we have demonstrated quite solid progress in getting rid of the porcine endogenous retrovirus, and we are making progress in addressing the immune and functional compatibility. And all of them wouldn't happen with a wonderful team. So this is a team at Qihan, and also we have close collaboration with different hospitals and agriculture universities, and trying to doing this together. And thank you so much for your attention. I'm happy to take any question if you have.
Hi, Stephanie Moore. I'm at Harvard Medical School. So I have a question for you. You talked about virus transfer from the pigs into humans, but presumably once the pig organ would be in a human, there might be human elements that they could then affect the pig cells. Is that true? And if so, what implications might that have?
That's a wonderful question. So while we are trying to create a tolerance environment to fool the human immune system not to recognize pig organ, we also create a situation or possibility that the organ will become the reservoir for cancer or virus to replicate without the immune surveillance. So we are quite mindful about that. One thing we are hopeful is we didn't touch the human immune system. Even the pig organ wouldn't recognize by the host, but hopefully the antigen will be presented by the human cell in the lymph node and still serve the function to clean it up. And another point I want to bring up is the risk-benefit analysis. So possibly for some patient, they are in very urgent need of organ transplantation, like cancer patient, and whether there are some scenarios, certain risk can be tolerated if it's life-saving. Thank you.
I have two, I guess, perhaps related questions. One is, after you've eliminated all the incorporating viruses out of the pigs, are they a little healthier than they were before, or the same? I mean, did it improve them or only make them the same? And then the second question is when you're testing this, because you don't necessarily test it with humans but do it with a primate, do you have to repeat this whole process for the particular primate as well as for the human, if you see what I'm saying, where you fix the antigens for humans, you have to fix them for monkey or something like that?
It's a very thoughtful question. So for the first one is whether there is some abnormality of PERV knockout pigs. It's still early day, it's 2 years. It's equivalent like to 30-year-old human. So we don't know yet. And it's actually an interesting scientific question. People know that for all the mammalian species, we all have endogenous retrovirus, whether they are like parasite or they are necessary evil. And there are some hypotheses. For example, people think they are important for intelligence, and people think a certain part of the virus is critical for placental development, thus fertility. And also people think that prime, the endogenous interferon, the immune system, for our body to fight against external pathogen. So for the fertility part, we don't think it's an issue because now we have the second generation, and the litter size, fertility, we don't think it get impacted. Intelligence, we don't know, so we try to look at how people check the intelligence of the pig. So there are some maze assay on the swimming pool, and when we try to put the pig in the swimming pool, they all have diarrhea. So we figure it's probably not important for us, but it's an interesting scientific question. And for the viral resistance, so far we haven't seen any abnormal immune profile of the pig, or they are more susceptible to infection, so we don't know yet. For your second question, whether monkey is an ideal model, because imagine if you put a monkey organ into a patient, there's still some rejection, but it's the best model we can find. And people recognize it's not a perfect model. Maybe the bar is too high or too low or totally different. So through our interaction with CFDA, China FDA, and FDA, we are quite encouraged that they are quite open-minded and data and science-driven. They understand the mismatch of our animal model, and actually we supplement those major models with other models like in vitro assay, ex vivo perfusion, for us to have the best understanding about the safety and efficacy. As long as we can justify some of our rationale and understand the caveat of different model, I think to the level we are confident. So we wouldn't do anything before we are confident about different models and the outcome of different models before we think it's safe to put into the patient.
Thank you.
Is there any way of estimating how many genes you will have to change in order to have an acceptable reduction of immune responses?
So the gentleman's question is there a way to estimate how many genes need to modify to make the organ human compatible? I think so. There are 30,000 genes in pig genome, so as human genome, and there are about 30% difference in terms of our peptide difference. So that, as you can imagine, it's a diminished return. In addition, we are not only getting rid of the antigen, we are also inserting some of the human immune modulatory factors. So there are two forces to make it compatible. Based on our experience so far, we don't think there are hundreds of modifications required. Maybe a dozen of modification could be sufficient as a first generation. Of course, there are innovation and improvement opportunity for future engineerings.
Hi. This is a very exciting topic and great talk. I was just wondering that when you modify and try to humanize the pig organ, do you have to do that to like the embryonic pig, or can you do that to an adult pig?
So the process we are running is actually doing modification on fibroblast.
Okay.
And as people clone DALI, we take the nucleus of the fibroblast, insert into a denucleated oocyte, then clone a pig.
Yeah.
So the reason why we don't do that in vivo on an adult pig is we want every cell and organ to be modified, whereas the in vivo editing efficiency wouldn't be practical to achieve that.
Right. So then how many years do you have to wait like for the pig to actually grow up and be a potential candidate for transplantation?
That's a headache for us because of the time constraint. So we do modification in the dish that can happen in 1 month. And we have to wait for 114 days, which is 3 months, 3 weeks, 3 days, for the pig to be born. Then we can do some assay.
I see.
But
Okay.
we do that staggeringly so that we are not waiting sequentially.
I see. And then just in the future, if you actually grow a pig to be an adult and then transplant the organ, you probably wouldn't want the pig to like have a growth hormone to make it speed up the growth process, right? So that it doesn't like modify the organ in any way. Like if someone has, if someone needs a humanized organ that's personalized, would they have to wait, I don't know, like 5 years, 10 years to get the organ from the pig? Is
I
that?
think that's a transformational part of our practice if we can make it work. So we imagine it's better practice than allotransplantation. You have to wait for the cadaver
Right.
or living donor. You can schedule that. So I can imagine that we have a big pig farm with good biosecurity. Then when a patient needs an organ, then we just harvest the organ, package it, then send to the hospital. So there shouldn't be any waiting time.
Cool. Thank you.
Hi, thank you so much for such a wonderful talk, and thank you so much for sharing this data. I guess I'm very curious as to how you guys are able to minimize the off-target effect of the Cas9 activity.
CRISPR is not perfect, as every one of us know. Among the 3 billion base pairs, sometimes it can hit the off-target. And in our practice, another toxicity associated with CRISPR is on-target toxicity. So imagine that once we modify multiple places, we actually effect multiple cuts in the genome. It can be healed in situ, but it can shuffle around and result in translocation. And this is correlated with the efficiency. The higher efficiency, inevitably, you will have more off-target and on-target toxicity. However, it's not a problem for our practice. So I can imagine it can be problematic if we put the tool directly into the patient as in vivo therapy or treat a population of the human cell, then transfuse back as ex vivo therapy. our practice is to treat a population of pig fibroblasts with CRISPR. Then we isolate single cell into single well, let them grow up into a clone. Then we QC part of the cell by sequencing so that we can identify the clone that with perfect modification with minimum off target, then clone a pig. In addition, we monitor the pathophysiology of the pig over time for us to better understand the long-term consequence of gene editing on a living animal. So that with the both safety got, we are pretty confident that even the tool is not perfect, our product can be perfect.
Okay, thank you.
I had a very naive question. Why the pig?
I think there are two questions in your questions. So the first one is, why not monkey?
Right? Or a horse or cow or...
So first, we know that HIV is passed from monkey to human, so there are higher chance of zoonosis. Because of that, to use primate as cell and tissue and organ donor is not allowed for safety reason. And why pig? So first, certain pig share similar size and physiology of human, so that it's easier to engineering and more size and functional compatibility. And the second one is, over the last thousands of years, we'll accumulate a lot of experience how to do a large-scale breeding of pigs in a controlled environment. You don't want your organ donor to raise in the wild. You don't know what's a pathogen, whereas the large-scale safety production of the pig is quite feasible.
So I had another question before. So iPS cells, right? Cells that are actually, you can turn into anything, right? Pluripotent stem cells. So how would they fit into this? I mean, you wouldn't need an animal to grow the organs if you were to use iPS cells.
I think it's also a competitive alternative approach. I think when it comes to cell therapy, maybe iPS has more advantage than Xeno. But when it comes to organ, I think the Mother Nature probably thousands of times smarter than us if we do a mental exercise how we can create an organ. So the first part is you have to have different components, hundreds of different cell types. The second one is how do you piece them together in a biological relevant way. And the third part is actually the hardest part, is how can you drill the vessel inside the organ to pump in the nutrition, the oxygen, and pump out the waste. I have to say that with the iPS be around for 12 years, we still have limited capacity to differentiate into those hundreds of different cell types, not mentioning step 2 and step 3. Whereas I think the beauty of xenotransplantation is it's a bioreactor with mature and functional organ. Of course, it has a baggage of xenogenicity that higher bar than human organ transplantation that we have to address. So I hope in the coming years, both approach can make concrete progress in order to deliver the medicine to the patient.
So the pig farms are, I'm assuming that they're very pure genetic background, right? Because they're sort of like mice. You know, in my experience with Black Sea mice, they're not from the wild, right?
Yeah, so they are good, healthy pigs to start.
Yeah, so they're very controlled. You basically control the genetic background, so there's no heterogeneity.
So unlike the mouse practice, it's actually a luxury in the mice field that people not only have inbred mouse, but they don't have the inbred deficiency, which demonstrate as lower fertility or lack of healthy status. And that's through hundreds of generations of breeding and selection. And as another student asked me, how long is gestation for pig? It's much longer, it's 4 months, so that there's no resources as mouse, which is inbred but healthy. For us, the way we control it is when we do the genetic modification, we want to make sure the founder lines have homozygous modification at the site we want. So even there's some diversity on the background, hopefully it wouldn't compromise the immune and healthy status consistency of our product. But it's a challenge issue for us, and we need to carefully QC the product to see whether we can achieve that.
Okay.
Hi there. You started your talk by mentioning the different options for transplant if it wasn't just harvesting more human organs, and the other option that you mentioned was bioprinting organs. So I had two questions. One, I was curious to hear your thoughts about, the viability of that option to grow human organs for transplant, and then also if the state of the science advances both for your technology and bioprinting, if you envision they could be complementary technologies or one would be preferable in the scientific and medical community to the other.
So I might be biased. So if it's a gamble, then I put all my chip into xenotransplantation for organ. As I mentioned, I think I'm pretty humbling after 10-year practice in bioengineering. Before CRISPR, actually, I spent years in the lab trying to engineer zinc finger to have new DNA binding affinity as a gene editing tool. It's very cumbersome. And until CRISPR came, and in my humble opinion, nobody invented CRISPR. Bacteria invented CRISPR. And we just repurposed that for gene editing. And similar concept here for xenotransplantation. I think it's so hard to create an organ we don't even understand the developmental process. That the beauty of xenotransplantation is there is a functional, healthy organ, but we just need to be smart in the way that to mark up the genetic makeup so that it wouldn't be attacked by the immune system.
Thank you.
Hi, that was a really, really inspiring talk. So I know you talked a lot about the genetic components of rejections and immunosuppression. Can you comment on the epigenetic aspects of organ donation in regard to accepting and rejecting? And along that line, the second question I have is, do you foresee in the future organ donation be sort of like a one-size-fits-all, where you can just grow an organ and then any patient, without regard to their genetics makeup, can accept the organ transplantation from pig?
I see. It's an interesting question. I never thought this way, like whether there's some epigenetic difference between the pig and human will trigger rejection. I know that some histone protein is recognizable by our antibody or T cell largely to clean up some of the damage of the tissue, because that's a time when the epigenetic markers will be exposed. I assume the protein are quite conserved between pig and human, but whether there's some difference and would be more immunogenic, that's a good question. But I think the pathway are quite conserved, so that at least I'm confident in the human host setting, there wouldn't be a problem for the proper epigenetic decoration while the cells in the organ are proliferating. And for the second one, how I envision the business model or commercial model. So first, my vision is to make the organ more accessible to the general public. So that entails two things. One is the cost, and the second one is the genetic makeup. So right now, we want to be an off-shelf product so that we can achieve the economy of scales to lower down the cost. For example, I just visited a farm which is supposed to be clinical grade pig farm, and the per diem per pig is $50. And imagine that if we are going to deliver a kidney from a 9-month pig to a patient, we need to raise the pig about 180 days in the farm, then that is about $9,000. So I'm quite excited about the possibility that we can create medicine, such a complicated medicine, with such a lower cost by using the bioreactor of pigs delivered to the patient. I can also imagine another scenario that we can customize the pig so that it's a better compatibility with the recipient. I can also imagine that in the future when we send the birthday gift to the baby, we don't send candy or toy, we just send a pig who can protect the baby for many years to come. I think that would be the next generation of product.
No more questions? Thank you, Dr. Yang. That was wonderful. It was very interesting. Thank you, everyone, and have a good weekend.